Now it's my great pleasure to introduce you to Kattie Weigh. Dr. Kattie Weigh is a Postdoctoral Research Fellow in the New South Wales DPIRD Soils Research Team based in Wollongbar, northern New South Wales.
Everyone, thanks for joining in today. So there's been huge interest among growers and the community to understand soil biology for the benefit of soil function, agricultural production and resilience. But there remains little consensus on which variables to measure, how to interpret results, and how to integrate results into soil health assessments in a meaningful way. So there's a range of tests we can explore to characterise a healthy, biologically active soil, and to compare the effects of management, amendments, and interventions on soil performance. So we've been developing a framework for the assessment of soil biology and how soil biology tests can be used to improve soil as well as make agricultural decisions. On the left, you can see where it all starts, and that is the microbial food source. The food source is mainly plant-derived carbon substrates. Then comes who's there, which reveals the abundance and diversity of bacteria, fungi, nematodes, and other organisms present. These organisms have the potential to compete and perform various functions. These potential functions do not always translate to what's actually happened in soil, particularly over time and with temporal fluctuations. This takes us to the assessment of actual function that has been performed. So after we've understood who's there and what they can do these food sources, microbes, and functions translate into agricultural outcomes like plant health or soil resilience? These outcomes then influence how effectively food sources are processed, completing the cycle and reinforcing soil health, which takes us back to the first component of the framework. And so the cycle continues. Throughout this presentation, we'll examine each component of this framework and discuss how they interconnect to support soil health. So as mentioned, there's a huge range of biological indicators that can be analysed within each of these framework components, all with varying accessibility, costs, standardisation, sensitivity, variability over time, and interpretability of results. You can find these outlined in the downloadable handout. So yeah, there's trade-offs, and we usually can't measure everything. And it's usually also not necessary to answer a research question either. So let's start by exploring the microbial food source. The primary source of food for microbes is soil carbon. Through photosynthesis, sunlight is captured and redistributed by green plants to other life forms, not only via fresh plant residues, but plants allocate up to 40% of their photosynthetically fixed carbon below ground, feeding soil microbes and initiating carbon cycling in soil. The root zone, which we call the rhizosphere, is a hotspot of microbial activity. Microbes cluster around these roots because plants release carbon-rich compounds known as root exudates that serve as food. The amount and type of root exudates shape microbial communities, attracting helpful partners like mycorrhizal fungi and deterring pathogens. The squiggly plot, or chromatogram here, shows some example chemical signatures of root exudate compounds. This plant microbe chemical communication has evolved over hundreds of millions of years to enhance both plant and microbial fitness. We'll explore a neat example of a beneficial interaction later in the presentation. So carbon enters the soil via plant residues, root exudates, or organic amendments in managed systems. And these exist as a continuum from fresh inputs to more stable forms. Particulate organic carbon, POC, is the readily available lay-bar pool, including plant fragments and partially decomposed organic matter, which can further be divided into free POC, which microbes can quickly decompose, and occluded POC, which is physically protected within soil aggregates, slowing its breakdown. Mineral-associated organic carbon, MAOC, is the stable, slow turnover fraction, consisting of microbial metabolites and necromass bound to mineral surfaces. Pyrogenic organic carbon, PYOC, is fire-derived, highly recalcitrant, and persists for hundreds of thousands of years due to its resistance against microbial decomposition. These percentage contributions are national averages estimated by mid-infrared spectroscopy, which is a rapid and cost-effective alternative to traditional physical fractionation for assessing these carbon pools. It's important to know that every soil has a carbon ceiling, which governs how much carbon a soil can store, which is influenced by soil type, climate, and management. For example, here we can see the strong relationship of soil organic carbon governed by clay content of the soil, where a greater amount of clay in a soil enables greater soil organic carbon accumulation, likely via the organomineral associations in MAOC, as well as the occluded organic carbon within soil aggregates. While some large-scale factors like climate and intrinsic soil properties like soil structure cannot be altered, we can improve soil structure through management such as the addition of organic carbon inputs. For example, biochar helps soils store more carbon by raising the ceiling for soil organic carbon storage. In this study, my colleagues used 13C isotopic labeling of carbon dioxide to trace carbon from plants into the soil after adding biochar. As plants absorbed this labelled carbon dioxide, their tissues and root exudates were also labelled, making it possible to track new plant-derived carbon. They found that in the biochar-emitted soil, the plants released 20% more root exudates into the rhizosphere, and microbial mineralization of organic carbon slowed by more than 5%, inducing what we call negative rhizosphere priming, all of which resulted in more new carbon stored in the MAOC, our stable mineral-bound carbon. Together, these changes mean that biochar not only adds persistent carbon to soil, but it also helps soil stabilize new carbon by modifying plant-microbe interactions. So now that we understand carbon food sources, let's explore which microbes consume these resources. So soil is the most alive and diverse ecosystem on planet Earth, with a gram of soil harboring up to 10 billion microbial cells, representing hundreds of thousands of different microbial taxa. Molecular tools have evolved rapidly over the past few decades, and there are now powerful tools to assess soil microbial biomass and diversity. But method selection should be based on research question, resources, and the level of detail required. So microbial biomass carbon makes up 1 to 5% of soil organic carbon, and it's influenced by a range of factors, including soil moisture, pH, as well as the amount and type of carbon in the system. In a well-known global biogeographic survey by Fiera and Jackson, soil pH was found to be a master regulator of soil microbial biomass and diversity. During my PhD, I quantified microbial abundances along the Hoosfield acid strip at Rothamsted in the UK, which is a very unique field gradient from about pH 4 to about pH 8. This allowed the effects of pH on soil microbes to be determined without confounding factors like different soil types and climate. This plot shows the pH sampling points collected along the gradient. And here we can see the neat relationship of bacterial abundances increasing linearly with soil pH. Fungal abundances exhibit a polynomial trend within this pH range, with abundances peaking between around pH 5, pH 6. And finally, the fungal to bacterial ratio of these abundances declined within this pH range. Here I used what's called quantitative polymerase chain reaction to count bacterial and fungal genomic regions and use them as proxies for their abundances. It's important to note that bacterial 16S copies don't equate to the number of bacterial cells, as bacteria can have up to 15 copies per genome. However, this method is fine for relative comparisons. Alternatives for quantifying microbial biomass include microbial biomass carbon, which is rapid and simple, but it cannot distinguish between biomass associated with bacteria or fungi. And then there's PLFA analysis, phospholipid fatty acid analysis, which is a gold standard in research, and it uses the cell wall lipids as biomarkers to reliably measure total bacterial and fungal biomass. In terms of characterizing the diversity of microorganisms, huge advancements have been made in this field in just the last few decades. With only about 1 or 2% of microbes able to be grown in a lab, the development of culture-independent methods, such as DNA sequencing, has not only enabled the Earth's microbial diversity to be characterized, but it's also enabled profiling of whole communities. Briefly, DNA is extracted from the soil sample, then key genetic markers, specifically bacterial 16S and fungal ITS regions, are amplified by polymerase chain reaction, sequenced, and then matched to known organisms using reference databases. Here's a neat example of how DNA sequencing helped reveal which microbes were recruited by plant root exudates. As I described earlier in the presentation, plant root exudates are key drivers for selecting and controlling who's found at the root surface. In this study, plants were grown under phosphorus sufficient and phosphorus deficient conditions. Their root exudates were captured and used as bait to see which soil bacteria they attracted. In the first figure, bacterial diversity is plotted by treatment. We can see that plants grown under P deficient conditions attracted a less diverse cohort of bacteria. The second figure shows a breakdown of who's there at a course level by treatment. P deficient plant exudates attracted many more bacteria overall, especially bacilli from the Firmicutes group. Now, if we zoom in further, you can see that Penibacillus, which is a phosphate solubilizing bacterium, dominated the community, attracted by P deficient root exudates. This population made up a remarkable 64% of the total, but it was actually just a minor player in P sufficient and control treatments. So this study highlights how plants can use chemical signals to elicit specific microbes, like Penibacillus, to help them overcome nutrient stress, but it also exemplifies the power of DNA sequencing and experimental design in deciphering these interactions. While DNA sequencing can reveal remarkable insights into microbial communities, such as those linked to management or amendments, this is what a raw DNA sequence file looks like. An important caveat to using DNA sequencing for routine soil health monitoring is that translating this data into human readable form requires technical expertise. And even then, simply knowing the names and identities of microbes may not clearly inform management decisions, which highlights the value of other biological indicators. So the focus is shifting from taxonomy to traits. That is, from just identifying who's there to an understanding of what functions those microbes can perform. Methods like qPCR let us target and count specific functional genes of interest, and it also allows us to count agriculturally relevant microbial groups. And in research, there's a shift towards employing functional metagenomics, which sequences all genetic material in a soil sample. This allows functional genes to be characterized and the functional potential of the soil to be estimated. In the interest of time, I won't go into any examples of functional metagenomics, but I'll touch on some more routine and accessible tests of soil function soon. So just like the example earlier, where I used qPCR to count bacterial and fungal abundances along the pH gradient, it can also be used to target and count specific genes as proxies, allowing us to assess the abundance of key microbial groups or functions. SARDI in South Australia provides the Predicta B service, which uses qPCR to count agriculturally important populations, such as mycorrhizal fungi, fungal pathogens, and nematodes. This sample report shows how these DNA-based counts are converted into crop-specific disease risk ratings, often visualized as traffic lights. Now, this is one of the few biology tests that directly supports agronomic decisions, helping growers choose fungicides, crop varieties, or even whole crops based on disease risk. While knowing who's present helps diagnose disease risk and loosely links to soil function, understanding what the community can do requires different approaches. Here we'll explore some methods to evaluate the functional potential of a soil. That is, its ability to carry out important processes like nutrient cycling and organic matter breakdown. These techniques allow us to profile not just who's there, but what the soil microbial community is capable of doing. Enzyme activity is closely related to important soil quality parameters, such as organic matter, soil physical properties, microbial biomass, and activity. Here we can see some examples of enzymes that are commonly measured, along with the nutrient cycle that they are associated with. Enzymes respond to soil management changes long before other soil quality indicators change or changes are detectable. For example, they tend to respond more quickly to practice change and other soil conditions compared to other soil health indicators, such as carbon content. However, they do bounce around a lot, often making it difficult to link changes directly to management practices. For example, in this field trial, we compared 2 organic amendments over 3 months, but enzyme activity was mostly influenced by moisture and temperature, not the treatments themselves. The large error bars in these plots showing enzyme activities by time also show how much enzyme activities can vary day to day. For instance, measurements at 10 a.m. on a Tuesday versus 5 p.m. on a Wednesday could vary substantially. Because of this high temporal variability, enzyme assays may not be the most reliable biological indicator for management decisions, but they could be paired with more stable indicators like soil protein, which we'll touch on later, or carbon fractions. Microbial respiration is another measure of soil biological activity, specifically mineralization. A key soil function is how much carbon microbes store as biomass versus how much is lost as carbon dioxide. The ratio of microbial biomass carbon to respiration, called the metabolic quotient, can indicate microbial stress or soil disturbance. For example, soils with copper fungicide residues often have a higher metabolic quotient, similar to how a person's heart rate may rise under stress. In contrast, healthy, more mature soils use carbon more efficiently, storing more and respiring less. Microbial respiration can be measured in field, as seen here, using chambers fitted with a septum from which gas is sampled, and carbon dioxide, nitrous oxide, and methane can be measured in a lab using GCMS. Substrate-induced respiration measures microbial activity in response to new carbon soil additions. This technique tracks how carbon inputs are utilized or allocated by microbes. In this example, soils received biochar and 13C labeled oxalic acid, which is a common root exudate. Carbon dioxide evolution was captured and measured using alkaline traps, which were changed throughout the experiment. In this cumulative respiration plot, the iron-enriched biochar treatment emitted less CO2 than other treatments, indicating greatest carbon stabilization. Isotopic tracing of oxalic acid using the 13C label then allowed partitioning of respiration into native and added carbon sources, as well as the quantification of new carbon in MaOC and POC. There are also commercial substrate-induced respiration assays, such as BioLog and MicroResp, that are used to understand the soil functional capabilities by measuring respiration responses to a range of substrates. These substrates range from simple sugars, which generally promote bacterial growth, to more complex compounds that may reveal activity of soil fungi. Both methods use colorimetric plate assays to assess microbial community function. It's available through Solvita or other accessible tools that quantify respiration, ammonia emissions, and compost maturity using color-reactive indicator strips, allowing rapid assessment of soil biological activity without the need for lab equipment. The teabag index, cotton strip assays, and the soil Urundi's tests are other easy, accessible tests that involve burying organic material for a couple of months and digging them up to assess decomposition, giving an indication of soil biological activity. The more degraded the teabag, strips, or undies reflects a more biologically active soil. These may be useful techniques to compare between sites, treatments, or management interventions. So these methods assess potential for microbial activities. The next part of the framework is soil function. So moving from functional potential to actual outcomes. Let's look at soil structure as an example here. Research has shown that aggregate stability is positively correlated with both soil organic carbon and soil protein, such as glomalin-related soil proteins. These relationships suggest that soil organic carbon and protein concentrations could serve as proxies or biological indicators for soil structure and function. Improved soil structure enhances water and nutrient retention, provides microbial habitat within aggregates, improves aeration, and promotes soil organic carbon stabilization. So in our lab, we are using this custom designed system to integrate biological indicators with isotopic tracing by delivering continuous 13C enriched carbon dioxide via a purpose-built gas mixer into a standard controlled environment chamber. The 13C label allows carbon to be traced from the plant tissue into the soil fractions, like particulate organic carbon, POC, and mineral associated organic carbon, MAOC, and even into microbial DNA. Microbes that assimilate the labeled carbon produce isotopically heavier DNA that can be separated to identify microbial guilds that are involved in carbon stabilization. This system also offers the opportunity to produce 13C labeled biochar by pyrolysing labeled plant material for use in future experiments. Going back to soil ACE protein is measured by autoclave citrate extraction, followed by total organic carbon analysis. It represents a substantial component of soil organic carbon, up to 40%, and it also represents a substantial component of total nitrogen, averaging around 28%. However, it's often overlooked in soil health assessments. ACE protein consists of microbial byproducts that bind soil particles together, enhancing aggregation, water retention, nutrient cycling, and microbial habitat. As the largest organic N fraction in soil, it degrades to amino acids and is subsequently released plant available ammonium. So in this study, planted systems produced more soil protein than unplanted systems, likely due to the additional carbon inputs from plant root exudates. Pending analyses for this study include 13C isotopic signatures of soil protein, DNA sequencing of microbial communities, and measurements of total organic nitrogen and carbon. ACE protein is emerging as a reliable indicator of soil health and organic nitrogen status. It responds sensitively to both long-term and seasonal management changes. It's also been linked to improved agronomic outcomes, such as higher crop yields. Measurement of soil ACE protein uses standard lab equipment, an autoclave, centrifuge, and a TOC analyzer. It's cost-effective, standardized across labs, and increasing knowledge about its role in soil is being realized. The final step in this framework is translating biological indicators into measurable agricultural outcomes, such as nutrient availability, water use efficiency, disease suppression, crop yield, agroecosystem resilience. Resistance and resilience are distinct but complementary properties that describe how soil systems respond to disturbances over time. Resistance reflects the ability to maintain functional stability during a disturbance, while resilience captures the speed and extent of recovery afterwards. Neither represents a single time point measure. So considerations for measuring soil resilience is that the monitoring offers dynamic insights beyond those periodic snapshots, but it requires intensive sampling with numerous replicates, highlighting the need for rapid, cost-effective soil monitoring tools. And there's obviously multiple stresses to consider, as well as the suitability of biological indicators to fit the need. Stresses including, say, drought, waterlogging, compaction, heat, pesticides, tillage, salinity, the list goes on. So our Biora controlled environment facility enables precision trials with high-resolution profiling of plant-soil interactions. The system provides temperature, humidity, and lighting control to simulate specific environmental conditions, such as a typical sunny day in Wollongbar, New South Wales. Automated water regimes use balance scales beneath pots to maintain target soil moisture levels. Our current priority at the moment is to identify biological indicators of success for overcoming various soil constraints, including soil dispersiveness, sodicity, acidity, as well as hard-setting soils. This system enables the isolation of constraint effects without confounding factors, the quantification of depth-specific water uptake, rapid amendment screening before field trial deployment, validation of microbial indicators against plant performance metrics, and the characterization of the mechanisms through which our amendments overcome these constraints. So finishing up, clearly there's a wide range of biological indicators that exist, all varying in cost, accessibility, sensitivity, standardization, temporal variability, and interpretability. But as mentioned before, trade-offs are inherent. Measuring everything is neither feasible nor necessary to address specific research questions. Strategic selection balances cost effectiveness with management sensitivity and robustness. So some emerging research and exciting developments include MAR spectroscopy, which could enable rapid cost-effective estimations of soil carbon fractions without the need for physical fractionation. DNA sequencing has rapidly advanced, offering high sensitivity, but requires specialized analytics and interpretation, and it is not always necessary for routine monitoring of soil health. And we've seen a shift from taxonomy to traits, that is, rather than which microbes are in my soil, to what functions are they performing. Soil protein is emerging as an indicator of soil structure, function, organic nitrogen status, and it provides good standardization at a moderate cost. Enzyme activities show high temporal variability, but they do quickly respond to changes in soil conditions. And great news is that commercial services are expanding, such as SARDI's Predicta B for quantifying disease risk and beneficial populations, and other tools and kits such as BioLog, MicroRasp, and Solvita kits to characterize soil function. So I'd like to thank you for your time today. I hope you learned a little more about soil biology, its complexity, and emerging indicators of soil biological structure and function. This is my team at DPIRD Wollongbar, New South Wales. Thanks again.
If you're watching this after the live webinar, you won't be able to access the handout, but please email us at the soils.unit@dpird.newsouthwales.gov.au email address, and we can forward that to you.